So today's video is going to focus solely on enzymes. We've talked about and kind of touched on enzymes a little bit in previous videos, so now we'll go a little bit deeper into them. Enzymes are considered a biological catalyst. A catalyst is going to be something that speeds up a chemical reaction. So a couple of reminders kind of before we get started. Remember, enzymes were a type A of protein, an example of a protein. All enzymes are proteins, not all proteins are enzymes. Remember, proteins could have a wide variety of functions, structure, hormones, etc. So because enzymes are a type of protein, remember they'll have an amino acid monomer, they'll contain nitrogen, right? That they're going to have characteristics of proteins. They are a biological catalyst in the sense that they're going to speed up chemical reactions. And in case you haven't caught from other videos, RxN is a reaction. So you can see in this graph here to the right, when we have a chemical reaction, just a quick reminder, let's say we have a synthesis reaction, right? So on the left side of the arrow, those are my reactants. And on the right side, that is my product. Regardless of whether it's a building up or breaking down type of reaction, right? Still product, still reactants on the left side. So if you look at this graph here, the reactant going in versus the product coming out, you can see there's this portion here called activation energy. Okay, so activation energy is the energy that is required to essentially get the chemical reaction going. And if you notice on this graph, when it's catalyzed, this hill is really tiny, right? So I don't need as much activation energy when the reaction is catalyzed. When it is uncatalyzed, you'll see this hill is very tall, and I need a lot of activation energy to get the reaction started. Think a little bit about like if you were about to run a race, right? And I needed to get to the starting line to even start the race. Would I rather run over a little tiny hill to get to the starting line or over a big giant mountain, right? I can end up running my race faster if I haven't used a whole bunch of energy to just get started. So chemical reactions can be sped up by enzymes because they're going to lower the activation energy. So that's how an enzyme is going to speed up a chemical reaction. It's going to decrease that activation energy. It's going to take that mountain and it's going to turn it into a little hill so that the chemical reaction can get started faster. So some common vocabulary that you will see as we talk about enzymes is going to be the enzyme itself. So in this picture, the enzyme itself is this purple thing. Okay, the substrate is going to be the molecule that is binding to the enzyme. So whether it's molecules as I'm building something up or molecules as I'm breaking something down, but that's going to be my substrate. The active site is going to be located on the enzyme proper. The active site is going to be where the substrate binds to the enzyme. And there's only going to be one particular spot where it can bind. So that would be the active site. And then the products would be whatever's left over. You'll notice as this enzyme has gone through this reaction, this enzyme was not broken down, right? The enzyme itself is reusable. It is not broken down during the reaction. It will, now that that enzyme has catalyzed this particular substrate into those two separate products, that enzyme is now going to go pick up another substrate and do the same reaction. Enzymes are substrate specific, meaning that they only bind with one substrate. So if I have, and let's say I'm talking about the enzymes that I use when I eat food, the enzymes that I use to break down carbohydrates. Those same enzymes do not break down proteins because the enzyme is substrate specific. The proteins wouldn't fit at the active site there. That active site would be made particular for the shape of carbohydrates, not for the shape of proteins. When the enzyme is able to bind to the substrate, it will slightly change shape. It's not a perfect fit. How we said previously that enzymes are substrate specific in the sense that the substrate only fits into one particular enzyme. It's a really close fit, and when the substrate gets there, the enzyme will kind of rearrange itself into what we call this induced fit model. So here's my active site here that's empty. There's no substrate in it. And you'll notice when the substrate is bound to it in the second picture, you notice how it kind of did a little bit, let me change that to a color you can see better, how it did a little bit of a change here in the shape of the enzyme, a little bit of a change here, just giving it a nice, tight, snug fit around that substrate. And so that is called our induced fit model, how that enzyme will kind of mold real nice and tight around that substrate. So enzymes are going to be a regulator of our metabolism. Metabolism is all of our chemical reactions of life. If you remember from previous videos, we talked about anabolic reactions versus catabolic reactions. Remember, anabolic reactions are those synthesis or building reactions. Catabolic are those breakdown or digestion reactions. Catabolic, catastrophe, right? Things fall apart during a catastrophe. So enzymes control all these different types of chemical reactions, regardless of whether they build or break down. So some specific examples that we've already talked about, right? So just reminders, these dehydration synthesis, right? I take the monomers, I remove the water, I use an enzyme, and now I've built this nice polymer. I've synthesized or made something. Enzymes can also participate, so that would be an anabolic reaction. Enzymes also can participate in these catabolic reactions. The example, remember the hydrolysis, right? I add the water to break, lysis is break, using an enzyme to help control this reaction. And now I end up with two monomers here. Together, all these types of reactions are going to be our metabolism, our building up and breaking down reactions all put together. So there's going to be a variety of environmental factors that can impact an enzyme's ability to do its job. We're going to look at some of those environmental factors, and then we'll also wrap up and talk about competition for an enzyme's active site. So let's look at what would happen to the reaction rate if I increase the enzyme concentration. So you see I have a graph here. Enzyme concentration as I move to the right here, that means it's increasing to the right, reaction rate increasing as it goes up. If I have a bunch of substrates, oh, why do I always pick hexagons to draw? So I have a bunch of substrates and I add more enzyme. That's going to speed up my reaction rate, right? It's going to speed it up though to a point. Eventually that reaction rate is going to level off and it's not going to be able to continue to maintain an increase in reaction rate. Essentially what will happen is all the substrate will be bound to enzyme. And it doesn't matter if I keep adding more enzyme. So all the substrate is bound to the enzyme. I can keep adding more enzyme. So while my enzyme concentration, so in this portion of the graph right here, let's make sure we're comfortable with graphs, right? So in this portion of the graph right here, my enzyme concentration is increasing, but my reaction rate, right, my slope of my line is staying the same because I've bound all the substrates busy. It doesn't matter if I keep adding enzyme, it's not going to speed that reaction up anymore. But at first, over in this section of the graph, the slope of my line is steep, right? So my enzyme is increasing as I move from left to right, and my rate, which is slope, is also increasing. So adding more enzyme should increase my reaction rate to a point. So what if I add more substrate? Okay, so if I add more substrate, so now I have a set amount of enzyme. Okay, I add more substrate. So again, my reaction will start to speed up, but eventually it's going to level off. It's going to become what we call saturated, where it doesn't matter if I keep adding more enzyme, all the enzyme that I keep adding more substrate, the enzyme is all busy. The enzyme can only work at a particular rate. Once all the enzymes are constantly being used, and remember, enzymes are reused, so use and then go grabs another one, and use and go grabs another one, that reaches that saturated point, and so it will level off as well. So increasing enzyme concentration and increasing substrate concentration can increase reaction rates to a point. So what if we change temperature? Okay, so temperature and pH that we're going to talk about are both they vary from enzyme to enzyme. Each enzyme is going to have an optimal temperature. So let's be clear that should be Celsius. These are typical enzymes in a human body. They're going to peak, and then they're going to start to go back down. Okay, so they're going to have an optimal temperature. Maybe it's a bacterium that lives in a volcano. Well, it would have a much higher optimum temperature. Maybe it's an organism that lives in the Arctic. Well, it has a lower optimal temperature. So the reaction rate when the temperature is too low for whatever that optimal temperature is. Okay, so when the temperature is too low, the molecules, think back to middle school science, the molecules move slower. Well, enzymes and substrates literally have to physically run into one another. So if the molecules are moving very slowly, then I'm going to have less collisions. So I have less collisions. Therefore, I have less enzyme reactions because I have less molecules, less substrates running into enzymes, right? As the temperature increases, what happens to the molecules, right? Well, the molecules, their movement speeds up. So I have more collisions between enzymes and substrates. Therefore, my rate will increase, but this is to a point. And we'll talk about that point in just a minute. But this is a really important concept that we need to understand with temperature, right? Temperature controls molecule movement. So when it's cold, they move slower. When it's warm, they move faster. Okay. I need for a substrate and an enzyme to have a reaction, I need them to collide with one another. Therefore, if they're moving a little faster, they're more likely to collide. So as a general rule, that will speed up my reaction rate. However, that's only to a point. And we'll talk about what happens when it gets too hot in a second. So as we mentioned, different organisms have different optimal temperatures. Like we mentioned before, this is a human enzyme. This one over here is a hot spring bacteria enzyme. So it can handle a much hotter temperature before it becomes too hot, before it hits that magic point where the enzyme won't work anymore. Okay, let's talk about pH for a second. So pH, again, organisms will have an optimal pH range. pH is essentially our hydrogen level. pH is dealing with acids and bases. It's a way to measure acid and base. We have our pH scale, because I don't think you may or may not have done this yet, right? So I've got 0 on my pH scale up to 14, right? 7 is neutral. So we treat 7 like your 0 point, right? So as I get closer to 0, I actually get more acidic. And as I get closer to 14, I actually get more alkaline or more basic. They are both equally essentially dangerous, right? Things that are alkaline or basic, those are things like ammonia, nitrogen-based compounds like ammonia, the bleach, right? So those are very dangerous, just as dangerous as any acid could be. So I'm afraid that some of that may have been cut off on the edge. So alkaline or basic as I go towards 14. So again, for instance, pepsin, this one's found in the human stomach. Well, we know there's acid in the human stomach, so its optimal pH is much closer to more acidic, right? To that lower, more acidic range. We've got others in our body that are more neutral. Trypsin, which is going to be more optimal at a slightly above neutral, more basic level to help neutralize the acid. Okay, so different enzymes have different optimal pHs. Essentially, what you need to understand is if they're not in that optimal pH range, they most likely will not work because they will denature. We're going to talk about what denaturing is, right? Okay, so let's talk about when we denature something, when we denature a protein. If you remember from our protein video, we talked about how the protein shape was critical to its function. And we looked at the picture with all the bins and folds and said, if these folds were missing, then it's not, a willy-nilly structure. They're not just folded however they feel like. If these folds were done differently, this protein would not work anymore. So in high heat and pH, major pH changes, the result is we break the bonds that control the protein folding, that essentially shape the protein. And when we do that, the protein loses its shape. See how it looks kind of sad and wonky, and how the active site isn't quite the right shape and it's losing its shape on the edge here? This protein or this enzyme has been denatured. If it has been denatured, it will not work. You'll notice this does not happen in extreme cold. Does not happen in extreme cold. This only happens in high heat and major pH changes. But that denaturation changes the protein shape. The protein can no longer do its function. Therefore, it won't work. For instance, this is what happens in like a heat stroke situation when our body temperature gets too high. Our enzymes can denature. Therefore, we can't control chemical reactions. It's why we get so sick. Okay, so we were talking about the enzyme being denatured and broken down, so therefore it doesn't work anymore. And let's look at another way that the enzyme could be prevented from working. So two different types of inhibition. So the first type of inhibition we have is what's called competitive inhibition. Okay, let's draw a very simple enzyme here, right? So I've got a very simple enzyme here, right? So my active site is right in here. Right? There's that active site. So remember, the substrate goes in and bonds to that active site. If I have competitive inhibition, so let's say here's my normal substrate, but now I have another molecule that looks a lot like that substrate. Well, that inhibitor can fit right here into that active site. So if that inhibitor bonds to that active site, it has outcompeted the substrate for that enzyme. So the enzyme the enzyme isn't bonding to the substrate, it's bonding to the inhibitor. So competitive inhibition, the inhibitor bonds directly to the active site. So physically blocking the active site from the substrate. in allosteric inhibition, there's my enzyme. Now I have an inhibitor that comes in bonds on the other side. And when that inhibitor bonds on the other side, what do you notice happened to your active site? That's a kind of a terrible. You'll notice your active site has now changed its shape, right? And so the substrate though didn't change its shape. So the substrate can no longer fit in that active site. So an allosteric inhibitor is going to bond somewhere other than the active site, but it's going to change the active site shape. So the little triangle means change. So competitive inhibition, I've got a direct inhibitor bonding straight to the active site. Allosteric inhibitor, I have an inhibitor that bonds somewhere other than the active site, but the result is it changes the shape of the active site, so the substrate still can't bond. Either way, these molecules inhibit the enzyme from doing its job. Okay, same as always, write your questions down because you may not remember them, especially if it's been over the weekend or something. Star by stuff you didn't quite understand what it meant, right? We'll spend a couple of minutes doing quick, answering quick clarification questions so that we can move on to modeling and a lab with this information.